Okay, so hi everyone. I'm standing here as the representative of the youngest team in ERG, not specifically by chronological age, but we've been around for about 8 years. So since I first joined ERG back in 2015, we've been building and growing as a team and building this research area as well, which really fuses two topical global challenges, so air pollution and plastic pollution. And today I'm going to give you an overview of that, a bit about the history, the advances that we've made as a team, and then a bit about the possible impacts of this newly recognised air pollutant. So just to set the scene, plastic is the third most abundant man-made material on the planet after concrete and steel. In 2015, we had produced an accumulative 6.3 billion tonnes of the material since its mass production began back in the 50s. And unfortunately, we're not very good at managing this waste, so 80% of that accumulative 6.3 billion tonnes had either been released to landfill or to the environment. And once in the environment, it obviously persists and accumulates, but it does eventually start to degrade upon exposure to sunlight, and it sheds these microscopic and nanoscopic particles that we know as microplastic. So plastics are essentially, at the molecular level, they're long chains of carbon, which we would refer to as an organic polymer. And obviously they're synthetic because they're man-made, and microplastics specifically would measure less than 5 millimeters in size in their longest dimension, and they may or may not contain additives or other substances. So plastic itself is a chemical, but it's non-volatile, it's solid and insoluble, but it also contains other soluble or partially soluble chemicals within it too, such as phthalates, antimicrobials, things like that. So microplastics occur in the environment as a variety of different shapes. You get fibres originating from synthetic textiles, you get fragments from the breakdown of larger plastic items, and even funny granular shapes like tyre wear, as we've heard mentioned in the previous talks. So this really reflects, I guess, the breadth of applications of this material across different sectors, different types of materials, but it also makes it a very complex pollutant to study. Because just by its size definition alone, it spans 6 orders of magnitude in size. And then you've got the range of shapes, the range of different plastics, and you can see it's really a class rather than a single pollutant. So just to give you a feel for the timeline of microplastic pollution, the term was first coined in 2004. And really since then, up until recently, it's been traditionally recognised as a marine pollutant, and that's because we first observed it in the marine environment. But as time's progressed and research has taken place, our understanding's grown to acknowledge that it also contaminates freshwater environments, terrestrial environments, but more recently we've realised it's in the atmospheric environment, it contaminates drinking water, food, and more recently, different human tissues. And so it's not hard to realise why it's on the radar as a potential public health issue, but really it is still growing as a field. There we go. It's growing very quickly. So as you can see, this is the publication rate, or the number of publications over time. And what you can see is there's a really steep increase. So it's really exploding as a field of research, and that's for both microplastic in general and microplastic in health. And I would say that as time has gone on, perhaps most of the data has improved in quality, but one thing we've constantly had to battle as researchers in this field is background contamination, because this is an ubiquitous contaminant. It's found in the modern laboratory environment, and so it's very difficult to keep microplastic out of samples you're analysing. And so this is something we're constantly having to battle with, which really does threaten the quality of data as we progress. So I'm going to focus on atmospheric microplastic. When I first joined ERG back in 2015, I actually came with an interest in oral toxicity of ingested microplastic, because at the time the only evidence relating to human exposure to microplastic was based on the contamination of shellfish. So a few studies had found that microplastic were in shellfish from the supermarket, from the shop floor, destined for human consumption. And so whilst I'd been looking at the impacts of microplastic in marine species for my PhD, I was in the knowledge that we were exposed, and that really triggered my interest in what that meant for human health. So I joined ERG because of the interest in particles, in toxicity. Those were the overlaps. And obviously surrounded by the amazing research going on in the team, I quickly became interested in whether this material was airborne as well. And so the first kind of challenge was to figure out, are they in the air? So we did some sampling on a rooftop in central London. We were then at King's College, so it was on the Strand campus with a very nice view. And we found that over the course of a month, as we sampled the atmospheric deposition that was settling naturally out of the atmosphere, we found microplastic contaminated every single sample. And this is 50 meters high off the ground. Just to put that into context, obviously there's a lot of error associated with this, so it's really just to get a feel for what the figures mean in terms of the numbers that we measured. It was equivalent to 2 billion microplastic particles depositing over the City of London borough every day, so for this area here. And as this is a persistent material, a big question is where is that going, where is that accumulating? So just for the bigger picture, the global context and where our numbers of what we were measuring fell, a few other studies have started to look at this issue, so in Paris, in China, in Germany, and in France. And these studies spanned different environments from megacities through to a pristine, remote mountain catchment in the French Pyrenees. And as you can see at the time, London came out of the highest deposition rate of 700 or so particles for a metre squared area every day. So as I mentioned, since then, the field's really started to grow, and now we've realized that this is a global phenomenon that's been observed around the world. So as you can see, the red tags represent observations of microplastic in air or deposition over a terrestrial environment. The sort of dark blue is for open ocean samples, and then the cream colors are microplastics in dust. And as you can see, this has really been observed around the world, and there are now more recent studies documenting it in Mexico as well. But up until recently, there's also been what I'm referring to as the size data gap. So the way in which we would detect and characterize microplastic was usually a microscope-assisted method. And the most commonly used method would have a limit of detection, so an instrumental limit of detection to what you could actually classify as a plastic down the microscope. And so as you can see, this limit of detection, or LOD, ranged from around 10 microns up to 50. So as of 2020, we were only aware of microplastic particles in the air or in atmospheric deposition or dust that were 10 microns or bigger. And what we of course know is that when it comes to human exposure in the airway, in the deep lung, it's the particles that are much smaller than this that are important for exposure. So these methods weren't capable of analysing the breathable fraction of microplastic. So another challenge we first had was figuring out how we could do that. So we used a technique called Raman microscopy. I'm not going to go into the intricacies of this. If you're interested, I'm happy to speak about it later. But essentially what this would do is, again, it's microscope assisted. You would focus your sample or particle of interest down the microscope, and you would generate a chemical fingerprint, as you can see up here, using the technology of the instrument. And then what we could do is we could also collect images, and for every pixel in that image, we could also generate a chemical fingerprint. So here you can see the chemical fingerprint of polyethylene terephthalate, or PET. That's a common plastic used in things like plastic bottles. And this is its chemical structure here. So this fingerprint relates directly to this chemical structure, and it's unique to these different materials. So then what we could do is we could look for specific unique peaks within that fingerprint and essentially map the presence of those peaks in our image. So every pixel where the peaks of the plastic are present fluoresce and show us a positive plastic signal, and the black means the plastic's absent. And so here you can see it's application mapping a polyethylene terephthalate fibre in an ambient air sample. So then we took this a little bit further, and this is the work of Joseph Livermore, who was doing his PhD at the time. He's now a postdoc in the team. And we wanted to really maximise the information we would use from that chemical fingerprint. And so Joseph generated some data, some analytical data methods to essentially map, rather than just mapping individual peaks, to map that whole chemical fingerprint in an image. And so as you can see, we could successfully differentiate between different plastics in a sample using this method. And crucially, if we were applying it to a sample that had been spiked with microplastic beads down to 2 microns, we could successfully differentiate them amongst the particulate matter. So this was a really important validation step to know that the method was doing what we wanted it to do, and importantly, that it was applicable to direct analysis in ambient particulate matter samples. So of course, then the next step was to apply that method to real-world samples, and as you can see here, this is just an example size distribution graph of microplastics observed in PM10 sample from Marylebone Road, so an urban traffic environment. So the size distribution really follows what you'd expect to see. We get a kind of modal size of around just below 10 microns. And here you can see this roughly coincides with our methodological limit of detection. So rather than the fact that the microplastics just stop at 5 microns, it's more than likely that there are smaller microplastics in the sample. They would just be on that limit of detection for that method at the time. And so in this particular site, we found around 2,500 microplastics per cubic metre of air, which, if you know about air pollution and number concentrations, is a relatively low amount, probably less than 1% or 0.1% of the total particle number concentration. But the methods do have limitations too, so aside from having this size cutoff, we also found that sometimes the chemical fingerprints were a bit messy, perhaps due to the presence of other impurities or coatings of the particles, and that really inhibited nice, clean classification. And so I would say that our numbers are definitely an underestimate for both of those reasons. So I'm just going to walk through another case study so that we've applied this working method to a few different studies now, looking at airborne microplastic in different environments. So here's an example from a study looking at indoor airborne microplastics. This particular environment was the university gym. We picked the gym because of the hypothesized release of microplastics during mechanical wear, from kind of wear and tear during exercise, from the machinery, from chemical plastic components of machinery and things like that. So we found, again, microplastic in every sample. Here's examples of microplastic fibers as opposed to particles, which relate to polyamide or nylon. As you can see here, we found microplastic in these samples down to sort of around 1 to 5 microns in size, and the different colours relate to different plastics found. And the majority were polyamides, nylons. We found some polyethylenes, polyamides, but really we hypothesized most of this was probably coming from the users. And the reason why we hypothesized that is because you can see this big error bar here in respect to our number concentration. So we found around 1,700 microplastics per cubic metre on average. We sampled over a 4-week period, and each week we saw a drop-off in the numbers. And funnily enough, we were approaching Christmas. So we just think that students were going home for Christmas in that time, and there was no one actually using the gym anymore. But you can see that, so that's why we kind of hypothesized it was probably coming from the local population density in that environment. And so just for comparison, you can see how this relates to concentrations measured in other indoor environments. So we have office and apartments in Denmark, we have university campus buildings in California, and residential apartments in Paris. So we've also done this outdoors as well, back on that 50-metre high rooftop in central London, so very much a background site rather than close to any sources at ground level. And we found very low concentrations there of around 2.5 particles per cubic metre, so extremely low. And you can see how that relates to other outdoor sampling campaigns done around the world. So that's kind of where we got to, and as I say, we noticed some limitations with the method. It was also quite low throughput, and whilst you collect large data sets, so those chemical maps or images literally would contain 1 million spectra in a data set. So it's high throughput in that respect, but it took a long time to collect that data. And so really for scaling up, for monitoring and aligning to the types of studies we wanted to do, it wasn't going to be a practical solution. And the other thing we really wanted to be able to do was to quantify the mass concentration rather than particle number concentration, so you could better align to some of the PM10 and 2.5 standard measurements that take place globally. So we've also started working on a different technique, or a complementary technique alongside. I'm not going to go into the ins and outs of it, but essentially it's a form of gas chromatography mass spectrometry, and it generates data sets like this, where each spot or kind of lit up part of this image relates to the presence of a chemical. So with this technique, you can literally detect upward of 1,000 chemicals in one sample. But for us specifically in this application, we were interested in whether any microplastic or plastics were present. And so all of these red spots with the labels each relate to a chemical marker of plastics. Some plastics have 3 different markers, some have 4, some have 1. And if all 3 are present, that really qualifies the presence of that plastic in our sample. And so as you can see, so this is again a PM10 filter that we directly injected into the instrument, or a subsample of a PM10 filter, again from Marylebone Road. And you can see here, for example, the presence of these 3 chemical markers which relate to the presence of polystyrene in that sample. And so then we've been busy working on how we would then lead to a quantitative method. We really need the aid of calibration curves. And so we've done a lot of work to optimize dissolving plastics in different solvents, creating these calibration curves. And so now we can derive concentration estimates as well for these samples. So moving forward, with respect to that line of work, our plan really is to apply both of those methods, obviously using the GCMS I mentioned as the workhorse, with complementary Raman as well. So we can now characterize and quantify different plastics, different shapes, different sizes in these important samples. And we're applying that in a range of different projects, some looking at outdoor microplastic pollution, some looking at indoor pollution. So this is an ERG-wide project looking at both the indoor and outdoor environments, and also in occupational exposure projects. So this is Joseph's fellowship here, looking at manual plastic waste sorters and their exposure in that industry. So I think the next kind of important steps as well, so we want to explore the relationships between plastics and plastic-related chemicals and other species, to sort of build on the hypotheses that they might be a carrier or from the same source as these chemicals. And then importantly, we want to start understanding key sources of emissions with some of the other teams in ERG. And so some of these sources might be sea surface spray, agricultural dust and sludge spreading, roads and city dust, landfill, and of course, plastics industry. So I'm just going to move briefly on to the impact. So why is it important to think about this as a component of air pollution? And is there anything particular about plastic that makes it different to any of the other components we know about? So once in the environment, plastic or microplastic particles, as I mentioned, undergo this weathering. This leads to oxidation of the plastic surface, which changes its chemistry. It might be coated in biofilms or proteins, and it might also gain contaminants that it accumulates from the ambient environment, like polycyclic aromatic hydrocarbons or transition metals, for example. So once inhaled, I guess one of the ways in which microplastic could theoretically cause harm is if any of those adhered chemicals or contained chemicals are released. So for example, if any metals are released into the lung lining fluid. And a big question here would be, does the amount of metal released from microplastic, does it exceed that which we're already exposed to via, say, an inhalation pathway, or is it just negligible? So is there a major contribution to metal in lung lining fluid from inhaling plastic alone? Sorry, not alone, but from inhaling plastic as part of what we inhale anyway. And then, of course, the other way in which they might cause harm is then through that direct interaction with cells, dependent on their size, depending on the cell type, and then other factors such as shape as well, and whether there's anything particularly harmful or of concern about microplastic compared to other particles. So this has kind of been explored in a World Health Organization report that I was a co-author on, which came out last year. It looked at in vivo and in vitro toxicology. We looked at occupational exposures, exposure via food, inhalation. But I just want to focus on some of the impacts that we highlighted from this. So some of the sort of key adverse outcomes to come out of these laboratory studies looking at the impact of microplastics were things like lipid and DNA damage, oxidative stress, inflammation, and interestingly, fibrosis as well. So tissue scarring in tissues in the presence of microplastic. But it's really important to highlight that the majority of these studies use monodispersed polystyrene spheres. So this was in 67% of the in vivo studies and 80% of the in vitro studies. And over 50% of those in vitro studies also tested nanoplastics smaller than a micron in size. And so there's a real kind of mismatch between the exposure we experience every day based on what we know from exposure data and then the hazard data that we're generating in laboratory studies. How can we extrapolate polystyrene spheres to, say, a fragment of weathered polyethylene terephthalate? And maybe it's fine, maybe it is, you can extrapolate, but we don't have the data to confirm that either way yet. So you're going to hear more about this, I think, from our next speaker, Todd, so just to flag that, who was the lead author on this report. So as mentioned, most of these studies use polystyrene beads, but if we look at all the different aspects of particles, you can see how we might overlook some of the issues with environmental microplastic. So we know shape, size, surface chemistry, surface charge. These are all important in particle toxicology, and they're not really covered in the tests that we've done so far, but equally we don't know much about the environmental microplastics in respect to some of these metrics. And so we really need to start thinking about more complex materials. And in addition, there's this extra layer then of environmental microplastics being weathered, having a microbiome on their surface, having additives inside, and then potentially having accumulated these contaminants from the external environment. So one of the reasons I think most studies have focused on polystyrene beads is that they are commercially available. So they're very easy to get hold of and to sort of start straight away. So one thing we wanted to do is actually start building a repository, if you like, or a library of particles that better mirrored or reflected those that we find in the environment. So with funding from the MRC, with the help of PhD student Erica Yang, who's on the Health Protection Research Unit for Environmental Exposures and Health, we've started building a library of different materials that are fully characterized, that are relevant to inhalation, and we'd predict they would deposit in the deep lung if inhaled, so they're relevant to pulmonary toxicity testing. And importantly, we've got different polymers and different shapes. So we can start asking really controlled questions just about the fundamentals before we even start to build the complexity of weathering, of coatings, of co-occurrence with different pollutants. So we can ask, does the type of plastic matter, or does the shape matter? So I'm just going to show some very preliminary data. This is from Eric's PhD, and it's really just the start of kind of the next 2 years of research. But essentially, we've started looking at that panel of microplastics in a couple of different cell lines and asking the question, is there any difference at all in terms of toxicity? So here, this is the data for an immune cell line. This is a measure of cell viability, which we could just, I guess, interpret as a metric of cell health overall. And we've got 2 time points, the light bars being 4 hours and the darker bars being 24-hour exposures to these different concentrations along the bottom. So what you can see is that at 4 hours, this particular particle, which is a polyamide particle, isn't really causing much toxicity, but at 24 hours we do start to see a decline in cell health. And just to remind you, the sort of lower the concentration where you see an effect, the more toxic that substance is compared to others that might have an effect at higher concentrations. And so if we're looking at the same particles in a different cell line now, so in this case it's lung epithelial cells, which we're doing in collaboration with Terry Tetley, you can see that actually we don't see much toxicity at all. and this is our positive control. So this is not a microplastic. I should flag at the end there. So already there seems to at least be different biological responses in different cell types, which makes sense. So then what about the role of shape? So this is just looking at one type of plastic now, but we're changing the shape. So we're asking, does shape matter? And these experiments or data I'll show you are for macrophages, so for the immune cells I mentioned. So here you can see for fibers, we do start to see a significant decline in cell health, viability, as the dose increases. But if we look at the polystyrene particles rather than fibers in the same cell, we don't really see any significant effect, which might suggest that shape, again, matters for this type of plastic in this type of cell. And then finally, just to show you what happens with the polyamide particles, so polyamide being nylon, in the same immune cells, but this time comparing it to a different type of plastic particle. So again, what you can see at 24 hours, we do see we see significant effects or drops in cell viability to quite a low concentration. Whereas when we look at the polystyrene equivalent particles, we don't see any effect. And so at this stage, we can't say why or what it is about these. It's just very preliminary data just to flag that actually looking at very simple microplastics might not be sufficient for capturing that diversity that we experience in the environment. So just to summarize, plastic sheds micro and nanoparticles and fibers. So I hadn't mentioned, but these are just some of the sorts of figures released in laboratory studies looking at wear and release from different types of materials for nanoparticles from plastics. They occur in air, but this is down to a size range linked to human health. But with the analytical challenge we face, our understanding is still very limited. They're persistent. We hypothesize them to be hydrophobic. This might change with weathering, we're not sure. But things like shape and chemical burden might also influence their degree of toxicity. Not all microplastics are equal, probably. And currently, this is obviously unregulated. So, microplastic emissions and release is not a regulated issue at present. And so the perception is that as plastic production, i.e. the source of microplastic, continues to increase, then the release of this contaminant will also increase. So where do we go? Well, I think we clearly need to be quantifying PM10 and 2.5 microplastics, to be building on the evidence base, and that's going to help us understand sources, but also potentially start looking at links to health outcomes as well. I think it's also important that we start thinking about what's driving toxicity and why, definitely in relation to what we experience in the environment, and we need to improve our methods to better understand nanoplastics as a fraction as well. So obviously this wasn't just me, this is the microplastic team. This is a bit of an old photo, but the core members, we've got Joseph, Eric, and Yukari. So I haven't mentioned Yukari's work on human tissue, that's another story. But I just want to say thank you very much to the team. We've also got a new member, Antonis, who's joined after this photo was taken. But thank you, everybody, and to all the collaborators who've helped along the way. Sorry.
Okay. So we've got a couple of minutes for some questions. There's one in the middle here. If we can get that across.
Thanks very much. That was really interesting. Do we know whether or not these microfibers and particles bioaccumulate, or are we very good at removing them from the body, or is it an unequal, unhappy balance between getting rid of them and picking some more up because they're so ubiquitous?
I would say there's not much data. I think the data that we have are from in vivo studies, probably looking at polystyrene beads, quite historical studies, just that were interested in particle kinetics in general. And there is suggestion that uptake and accumulation in different tissues does occur, but it's very much related to particle properties like size, and surface chemistry, and things like that. So no studies have currently looked at microplastic fibers, microplastic particles, different types of plastics, and whether they're taken up and where they'd accumulate. But it's very likely that size is the really critical factor governing that.
Okay, just one question here. It's the last question.
Thank you. Anne Hansel from the University of Leicester. I was just wondering what toxicological data are available on the chemical constituents, and if you're able to make any comparisons between what you're seeing in your in vitro study with the particles as compared with what's already known about the chemical itself.
So I'd say that, yeah, for chemical additives in plastics, there's a whole wealth of literature that have looked at the additives alone. So we definitely know, I guess, the hazard and the characterised hazard for them. What we don't particularly know is like the leaching rate from microplastics. In these studies, one of the things we're definitely going to follow up with is checking leachits and whether you see the effects due to the leachits or due to the particles themselves. So I think, yeah, whilst we know what the additives do, we don't necessarily know the rate of release and therefore the dose of those additives relative to the microplastic. And what's at least predicted based on, I think, probabilistic modelling studies is that at least based on the exposure concentrations that we know of so far, the levels are too low to have a kind of significant contribution to the overall body burden of those chemicals.
Okay, thanks very much, Stephanie. A great talk. Thank you.